This is Chelsea Holman, and I'm going to talk about the KIT lab activity that I chose, which was based on enzymes. The lab that I chose came from eScience Labs, which can be found at www.esciencelabs.com. It's one of the custom kits. Specifically, it's titled Oregon State Teachers Kit number 1. When searching for it, you look for KIT 9151. The lab that I chose from this kit was Biology Lab 8, which is the enzyme lab. Now, just a side note, I did have some difficulties when ordering this because the original instructions said that the lab that was included in this kit would be the cell respiration lab. However, when I received the kit, the cell respiration lab was not there, and instead there was an enzyme lab. So I thus chose to do the enzyme lab. And this was just one of many difficulties that I had with this kit. Now I want to start by talking about the Understanding by Design outline that we discussed in class. The first stage of the outline includes desired results. So the first being content standards, I said was the functional role and mechanism of enzymes. This is the main goal that would be in a standard curriculum. Understandings and goals. Enzymes are important and molecules that perform a variety of tasks, and there are many variables that control the activity of enzymes. And the student was asked to explore these different variables and also the different tasks that enzymes need to perform in living organisms. Essential questions that could be asked throughout the lab include, how does temperature affect enzyme activity? And more specifically, why does temperature? What's the mechanism that temperature is controlling? How does concentration of substrate affect enzyme activity? Do all living things have the same enzymes, and at what concentrations? Finally, the student objectives were included in the lab directions itself. So these are all direct quotes. Describe enzymes, structures, and functions, including activation site. Explain how enzymes affect activation energy of a reaction. Relate reaction kinetics, activators, and inhibitors to the function of enzymes. Hypothesize how climate-based changes can affect enzyme function. The next stage is assessment evidence. These would be tasks that we would ask the student to perform in order to assess how well they understand the concepts. The performance tasks include things like performing the experiments, recording the results, and analyzing the results. Other evidence things that are included in the lab are pre-lab questions, interpretation questions after each activity, as well as some more difficult concept questions after each activity. Finally, stage 3 is the learning plan, and these are the activities themselves. The lab that I chose had 2 activities. The first was activity 1, enzymes in food, and the second was activity 2, the effects of temperature on enzyme activity. The following are more detailed descriptions of these 2 activities. Activity 1 explored enzymes that are found in food. The basic setup for the experiment is to look for amylase inside different food products that are found in the house. And how they do this is amylase breaks starch down into free sugars. So by spraying starch onto a paper towel, that is then can be broken down by enzymes that are transferred to the paper towel from the food samples. The potassium iodine is used as an indicator for starch. It turns a very dark black color in the presence of starch and is just a light yellow when there is no starch present. These are the results I got when I did this experiment twice. On the top is the positive and negative control, as they called it. I disagreed with the terminology of positive and negative controls in this experiment. Their positive control was starch that was then sprayed with indicator, and the negative control was a paper towel that didn't have starch. Well, the paper towel that didn't have starch was a good negative control. An even better positive control would have been starch sprayed onto a paper towel, then treated with a purified form of amylase to see what our true positive result would be. The first time that I did the experiment, which is seen in the middle, I used 3 different foods: cheese, apple, and ginger. And here I didn't really see much amylase activity. And I found that this is actually because when I first prepped the paper towel with starch, I included far too much starch, and there wasn't enough amylase to overcome the levels of starch that I had added. When I reran the experiment using lower levels of starch, the cheese, apple, and potato this time all showed good results in terms of their ability to break down the starch. Some good things about this activity is the directions actually ask the student to determine what the negative control would be. Again, remember, this is the paper towel with the potassium iodine straight directly on it without any starch added. The directions were not really specific enough, however, because like I said, I had the problem with originally applying too much starch to the paper towels. They misused the terms positive and negative controls, as I explained before. I would have run 3 separate controls, and they were missing that purified amylase to show the students what a broken down starch result would look like. One of the other really good things about the lab, though, was that it could be repeated multiple times because there was way more in starch and potassium iodine than you needed. So, as I did, you can repeat it over and over again. Activity 2 looks at the effects of temperature on enzyme activity. In this lab, catalase, which was provided by yeast, was used to break hydrogen peroxide down into water and oxygen gas. The oxygen gas was then collected inside a balloon, and therefore the amount of reaction could be measured by the circumference of the balloon as it filled up. This was then performed at 3 different temperatures to see how the temperature of the substrate affected the overall reaction rate. The circumference was incredibly difficult to measure, I found, because as you can see in this image, the balloon did not blow up in a perfectly spherical fashion. Also, the way that the directions were written, it has you try to do all 3 reactions simultaneously and then go back and measure the circumference, all while making sure that each measurement is being done after 30 seconds of reaction. 30 seconds was not enough time for me to measure any of the circumferences. So what wound up happening was all of my data was for the end point, because it took me roughly a minute to a minute and a half to get all 3 circumferences measured. As you can imagine, if it's an endpoint reaction, there was a fixed amount of hydrogen peroxide, which means that all 3 samples had the same endpoint. So all 3 had the same diameter. So this did not work for me. It was then also frustrating because there was not enough sample for me to completely redo the experiment. However, I was able to visually tell a difference in how quickly the balloons blew up, as well as how much bubbling was formed inside the test tube during the reaction, so that the warmer samples were reacting more quickly than the cold samples. Overall, my impressions of both activities were not very positive. I was frustrated with the company and ordering of the kit. There was some complications in the original ordering process, and then once my kit was delivered, it did not have the same activities in it which I was told I was going to have. I thought that the topics of the labs cover high school level concepts and topics. However, the activities themselves were fairly simplified, and I would not have given to anything more than a middle schooler. It did have a great hands-on component. So for from a homeschooling perspective, this would be great because it does give that hands-on experience that you don't always get when your education is done remotely. The instructions I didn't think were written very well. There were a few errors, as well as a few misuses of terminology, which I found frustrating. I much prefer the traditional liver catalase lab that many people are familiar with, that looks at denatured protein or enzyme versus fresh liver, how increasing the surface area of the liver increases the available enzyme. You could also do this with different amounts of or different temperatures. However, I found the balloon concept was not consistent enough and I wasn't able to get the appropriate results. Luckily, I knew what the answer was supposed to be. However, a new student just learning this could have been misled. I also thought that the activities could have had a lot more detail in the concepts. The questions, with the exception of one or two, were all very simple in the end of the laboratory. And I thought that there could have been more difficult questions that really required critical thinking as opposed to just regurgitation of the previous intro material as well as the results. I also think that it's important to make more quantitative measurements, specifically with Activity 1. I would say overall that I would recommend this type of kit, probably not from this company, but I would recommend a kit lab to people who are doing remote labs. I just think that they need to be beefed up a little bit and more in depth so as to relate closer to what a traditional wet lab would be. Thank you very much for your time, and I look forward to hearing your comments.